You're listening to the FaceTime with Scientists podcast, where we go into the lab or into the field to learn from scientists about their lives, their work, their struggles, and their joy. Let's go. Well, hello everyone. Today I'm talking with Zainab. You're at the University of Georgia and you're a PhD candidate there, and I believe you're studying biochemistry and blood
Yeah.
immunology a little bit.
Yes.
Awesome. Well, I'm super excited to chat with you today. I've been talking to a couple other scientists recently on the podcast that work with organoids and stem cells and various kind of, you know, working on disease treatments through kind of stem cell and cell biology. So, kind of right in that vein, and I'm super excited to get your take today. Can you tell us a little bit about where you are and what you're going to show us today?
So right now I'm in the room that we keep our biosafety cabinets in, and I'm going to split cells. Not like literally split them, but I'm going to passage um B cells, specifically Raji cells. So they're B-cell lymphoma. They were harvested first in 1965, I believe. And it's just a cell line that's been going on and on. Usually cancerous cell lines can passage indefinitely because they grow so aggressively. So even though these cells are from 1965, I can still use them. I can follow my experiments. Sometimes they get too condensed and you need to give them fresh media. You need to lower the cell count a little bit. So I have to do it in this thing here, which is a biosafety cabinet. Um, it's supposed to keep everything entirely sterile. So everything that comes in needs to be sprayed with 70% ethanol. So I have my 70% ethanol here. Um, so I'll be using pipette tips, um, cell culture media, and you're ready to go.
Awesome. Yeah, super. Yeah, can you tell us, let me know before you start a little bit about, yeah, a little bit more about these cell types and what you're using them for. So, I mean, you're studying cancer in the lab. But yeah, what maintaining these cell cultures, what do you kind of use them for, I guess?
So for these ones specifically, I use them as a target cell. So what our lab does is we research natural killer cells, which is a kind of immune cell. And I'm focusing on seeing how best I can boost the natural killer cell's ability to kill cancer cells. So these are in target cell that I use in place of the cancer cells when I do all my stuff vitro. So like basically just in a dish.
That's super cool. Yeah, I'm super excited to learn more about natural killer cells, which they're called. They're like the ones that, right, just kind of go after everything and say
Yeah.
like, "Are you good or are you bad?" And if the cell answers nothing or "I'm bad," it kills it, right?
Yeah, yeah, yeah.
So, yeah, super cool. Yeah, it's so funny. I think, we always see scientists like or in the lab, it's like we're addicted to right, ethanol of like spraying
Oh
it
yeah.
everywhere. But yeah, so I mean, right, I think a lot of people when we see these hoods, it's like not maybe not clear that, sometimes, in some situations, there's something dangerous to the human in that, but in that case, it would be like higher biosafety conditions, right? But it's mostly like we're trying to prevent contamination, like of your cells.
Yeah,
Anything
yeah.
that would be going into the biosafety cabinet, you want it to be clean so it doesn't. Yeah,
Yes. So here I'd be preventing contamination of my cells because we just really want to make sure they're not contaminated to work properly so we can repeat our assays and things like that. And if they're contaminated, then they don't grow properly. There's something wrong with them. Um, so the biosafety cabinet hood also has like a certain airflow um, that prevents anything from coming in or well, mostly prevent stuff from coming in, like
right.
in terms of like air. So they'll test it with like a smoke tester, and you can physically see the smoke like being repelled from
Uh,
uh the inside, even when the hood is open. So that's why it has to be open to like this certain amount. So it's still below my head, but I can stick my hands in. So you can kind of see what I'm doing now.
yeah,
So my cells will usually, I'm not sure if you can really see it, but they tend to clump up into little balls.
yeah.
Um, so I'll sort of like very gently shake the flask in order to dislodge those so I can get an accurate cell count. I take out 50 microliters. So this is a P200, so 050 is 50 microliters. And you also have to be sure to keep with sterile technique. So I can't pass over anything that's open. I have a clean side and a dirty side. You can't see the dirty side, but I'm working on the clean side. So I'll get my tip. I'll make sure it's attached. And then I'll make sure that's all mixed up. And then I'll go in my flask, take out my 50 microliters, and I'll put it in my PCR tube. And then I pipette up and down just to make sure that my cells are evenly distributed. So I'm getting an accurate cell count. And then I take this and I take it out so I can go to my cell counter. And then I can bring everyone to the cell counter.
That's super cool. So, like, yeah, you're just right now, you're just seeing how many cells you have in a specific concentration.
Yes.
Gotcha.
So we have our cell counter. We have an automated cell counter, which is really nice because back in the day, you used to have to count everything by hand, which is not the funnest. So I
Yeah,
take
I
another.
remember that from my lab days of like, right, like taking something, putting it onto a like basically a microscope slide with a grid on it and under the microscope and then counting in a certain grid how many cells there were.
Yeah, I'm familiar. I'm all too familiar with hemocytometers. I'm so happy that I don't have to do that anymore. Okay, so I use this dye. I took 7.5 microliters of this dye called Trypan Blue. So it's a blue dye and it'll basically stain cells that are dead because their membrane has holes in it. So the dye is big enough that it can only go into the cell when the cell is dead, the membrane has holes in it. Otherwise, it can't cross the cell membrane into live cells. So I can count how many cells are alive based on how many cells are not stained with Trypan Blue.
Gotcha.
Okay. So I'll pipe that up and down my cells just to make sure they're equally distributed. I'll mix that with the Trypan Blue. Then I'll put it into these nifty counting slides. There's 2 sides. These are sometimes the bane of my existence because I have to pipette it in a very certain way to make sure it covers the whole slide, or else I have a bubble and I can't count my cells accurately if there's a bubble in my slide.
Right. Are you looking for like cell viability, or are you just kind of looking for like to inform you of when you'll need to passage or how much you'll need to dilute?
Because I've used these cells for a while and people have used these cells for a while, I know when they need to be passaged. I usually passage them every Monday and Friday. So every 2 to 3 days you can get away with passaging them. Start. But I want to know how many cells I have, and then so I can dilute it from there because we culture them at a certain concentration. Um, so we passage them at a certain concentration. So let's say usually I have like 1.5 million cells per mil in the flask, and I want it to be at like 300,000 cells per mil when I passage them. So then they still have more room to grow in the fresh media. So I have to do the math to count it that way. I forgot my
Right.
little book, so I'll be writing my math on this paper towel.
It's real science happening.
I'll transfer it over. It'll be fine.
As long as the paper towel doesn't get wet.
Yes. I am right next to the sink, but I'm hoping it doesn't.
When you're handling your cells in the lab, cells don't observe holidays, right? Like you have to
Yeah.
go into the lab all the time, passage your cells, like take care of them. Is it, I don't know, do you think of them fondly or like, oh, I gotta take care of my cells again?
I I know people that think really deeply about their cells and they cherish them. Um, I never do, and I kind of feel bad because like there was a guy in 1965, and this is like his B cell lymphoma, and he
Uh,
did donate these cells for the cause. So I should probably think of them like that. I just, I just don't. But I do. I like when my cells are growing. I like when they're doing the right thing. They're they're growing properly, and it seems like these ones are a little. They're a little sad, but I think they'll be okay for when I need them.
I think, right, like you can passage some of these cancer cells right so many times before they start to maybe not be so good for your experiments anymore, right? Or they start to get
Yes. more
more damage or that kind of thing, and then you have to kind of go back to your stock that's been frozen, right? It's been,
Yes.
yeah.
Oh no.
It's been a while since I worked in the lab with, I mean, HeLa cells, which were very,
Okay.
very hardy.
Yes. These ones are Raji cells. So they're still kind of hardy, I think. But I haven't ever worked with HeLa cells, so I have nothing to compare it to. Um, I think these will be okay. They're nearing their end of their passage. So like the ideal amount of passaging.
Right.
So they're getting a little finicky for sure, but I think they'll be fine. Um, so I'm at 1.27 million cells per mil, and I want it to be at 300,000 cells per mil in 10 mils, so 3. And then I just do algebra because the total count is 12.7 million. Math still comes back, you guys.
It does. I mean, I would need a calculator for sure.
Oh no, I'm still using a calculator.
Okay.
I'm just doing the cross multiply. Um, so then we'll go back to the hood and I'll take out however many cells that I need. And then I'll spin it down in the centrifuge so then I can add the fresh media.
Gotcha.
So now we'll move back again.
Nice.
So basically I treat my natural killer or NK cells with like this specific drug treatment. Um, and I'm trying to see how increased the killing of these target cells are with the drug treatment compared to without the drug treatment.
Gotcha.
Now my hands are also covered in ethanol,
Nice.
and I'll be moving however much I need. I usually label the tube with the volume amount so I can remember when I have to move it over. So this isn't proper labeling, like I'm not labeling it with the date or my initials, but I'm only doing some quick passaging, so it should be fine.
You were explaining like what the experiments are. What are you working specifically in terms of looking at ability of the natural killer cells to kill these cancer cells, like under different drug conditions you were talking about, and yeah, can you talk about like what those are? From my understanding, it's like the immune system already is primed to, should be killing our cancer cells, but for a variety of reasons, cancer cells might hide from the immune system, or um, our immune system might be like quieted down by the cancer cells in terms of not attacking them, right? So like kind of broad strokes, I guess. Yeah, what is your research hoping to achieve in terms of like helping immune cells kill cancer cells more effectively?
Let me just throw this in the centrifuge and then I'll answer that question. Okay. So basically, there are two different ways that natural killer cells can kill any sort of pathogen. Well, specifically talking about cancer cells. So there are two different ways that natural killer cells can kill cancer cells. And one of them is through a host of activating and inhibitory receptors. So it's kind of like there's a ton of different activating and a ton of different inhibiting receptors. And this is where the non-self theory comes in. So NK cells will basically kill anything that's not part of the body. So it has to express like a specific MHC. It has to express MHC. I think it's MHC1 is all cells in the body, and MHC2 is just immune cells. So if your body, if those cells do not express an MHC marker, then the NK cells know that, okay, this is not part of the body. And there's a ton of different play that goes into it. Like there can be other receptors, the inhibitory receptors can be partially inhibited, but then some of the activating cells can be activated. But it's sort of like, it's kind of like a two-sided scale. Like if you have enough inhibitory receptors and you don't have enough activating receptors activated, then it'll be inhibited and then the NK cell won't kill that. And if it's the other way, when you have enough activating receptors activated and not enough inhibitory receptors, then the NK cell will kill whatever's not part of the body. And then the other way that NK cells can kill is through a protein called CD16A. So this is really fun because this is what makes NK cells partially part of your adaptive immune system and not just your innate immune system. So your innate immune system basically kills everything. It acts first. It kills anything that's not part of your body. It's not specific. Your adaptive immune system are like your B cells and T cells, where you have things like antibodies and you have things like specific receptors, and it recognizes specific epitopes. So NK cells through CD16A can bind to antibodies, and then those antibodies will be bound to specific epitopes on your cancer cell. So it sort of works together with like monoclonal antibody treatments for cancers. And with this process, it's called ADCC, or antibody dependent cell mediated cytotoxicity. And basically, the natural killer cell will kill anything that is attached to that antibody as long as it's bound through this protein called CD16. Does that make sense?
Yeah, I think so. Yeah. So, yeah, you have your natural killer cell. It has some receptor or something on it that attaches to antibodies, I guess, that are your body makes, or you would have to,
Yeah,
or these are
it's
things
either,
that could be drug added to the
yeah,
body.
yes.
And then if those attach to the cancer cell and also to this natural killer molecule, then you're kind of getting that match where the natural killer cell can now kill the cancer cell. So I'm guessing these antibodies, yeah, maybe if your body's naturally good, can it make them so that it's helping your natural killer cells recognize the cancer, but it could also be stuff that we're creating through drug trials.
Yeah, so there's a lot of monoclonal antibody drugs that are out there that are specifically for cancer. A big one is Keytruda. That one binds to, I think, PD-1, and that in a nutshell is basically like a immune checkpoint that cancer cells exploit. So certain cells will express PD-1, and then when it binds to the ligand, it's like, okay, I'm part of the body, please don't kill me. And now cancer cells are expressing the ligands, but also sometimes the protein, PD-1. PD-L1 is the ligand. So using this drug, it can block that interaction. And then because it's an antibody, NK cells can bind to the other side of the antibody and still facilitate the process of ADCC. So a lot of monoclonal antibody drugs are part of the evaluation on how effectively they work is seeing how well they do ADCC as well.
Gotcha. And are there certain antibodies right now that are in use for treating patients that are specifically for like binding to natural killer cells to get them to kill cancer
Yeah,
cells?
so I mean, like, I haven't read about every single monoclonal antibody drug, so I can't say this like consistently,
Right.
but I'm pretty sure that when they go through the process of like, okay, is this treatment effective? They also want to see how well can it do ADCC based off of just like compared to just like some like different antibody or dummy antibody or something like that. Because that's like that's very important. It's a very important part of the process. So like when they evaluate um these cancer drugs in humans, they also see, okay, how well are they doing ADCC in these people? So how well is it killing cancer in this specific path? There are other pathways of the immune system that can also kill cancer, but this way is also important, especially because it directly interacts with the antibody. Does that make sense?
Yeah, you
Okay.
were saying the ADCC, is that what
Yes,
you were saying?
ADCC.
And that specifically, sorry, can you explain that again? That is the pathway with the natural killer cells?
Yes.
Gotcha.
Explain the pathway again.
Well, yeah, like yeah, I just kind of forgot in the midst of that what the
No, it's
ADCC is.
okay. So you have natural killer cell, and then you have a protein on your natural killer cell. And then that binds to the antibody on the tail end. So like an antibody shaped like a Y. So this tail end is what's bound to the natural killer cell. And then that Y portion is what's bound to the cancer cell that you want to kill. So it's bound to like some sort of protein that's expressed on the surface of the cancer cell. And then once that connection happens, then the NK cell is like, okay, I'm killing that one specifically.
Gotcha, and that whole process is called ADCC?
Yes.
Gotcha.
I know it's a
Gotcha.
little bit confusing, especially because antibody drug conjugates or ADC is very popular right now. So lots of similar acronyms.
Gotcha. No, that makes sense. So you're saying that, a lot of antibody drugs, maybe they're kind of benchmarked against being able to kind of do this connection of bringing the natural killer cell basically to the cancer cells to kill them.
Yeah, I'm pretty sure that's part of like the evaluation process when they look at how effective these antibody drugs are.
Gotcha. That's super cool. Okay, so in your research, are you looking at kind of improving creating new connections there or yeah, kinda
No, so I'm trying to see. So I'm basically modifying the cell itself. So I'm modifying the protein that the antibody binds onto through this drug to see if I can like boost binding affinity because boosting binding, this isn't always the case, but it is the case specifically here that if you boost the binding affinity, it will also increase ADCC. My goal is that I want to boost ADCC because if you can boost it, um, then you can use less of the drug. Um, and that's usually pretty good because these drugs can be really expensive. Um, and then it can just like generally be a more effective treatment. You know what I mean?
right. That's
Yeah.
super cool. You would have to get the drug into cancer cells, right? Like, you would have
So
to
it'll be
modify
in
cancer.
the natural killer cells. So I'm trying to modify the natural killer cells. So I get them from people, their primary natural killer cells. So they come directly from people, no cancer, no nothing. And then I treat them with this drug combination and then to hopefully increase the amount of ADCC these NK cells can do, and then add them to the cancer cells.
Ah,
And then
gotcha.
evaluate that.
That's super cool. So, I mean, I guess ultimately we're hoping we could, um would we be, like, giving people natural killer cells that are modified, or we would be delivering something to people that would modify their natural killer cells?
So the idea is that it would be an autologous like NK cell therapy. So that means that you take out people's natural killer cells, like their own cells, put them in a dish, add this treatment, put them back in. Is
Gotcha.
the thought.
That's super cool.
Thank you.
Yeah, that's amazing. I mean, yeah, I mean, obviously the science is complicated, but that's super neat. Have you always yeah, what's your background? Have you always been interested in cancer research or
I think so. So it started way back in high school. I did this science summer camp for girls. It was really cool, but I don't think it's run anymore, which is kind of upsetting. But we got to tour a bunch of research labs. Um, and I'm from Pittsburgh, so it's really like lucky that there's like 7 universities there. So we got to tour a bunch of research labs at different universities, and one of them was studying aortic aneurysms, and I was like, "That's really cool. I'd love to know more about that." And they taught us how to network before we got there. So I was like, "Okay, I'm gonna use my new networking skills and I'm going to get the contact information of this person. And then I learned that like you can have high school students in labs. Now, as a PhD student, I know they don't do much, but like when I was a high school student, I was like, this is so cool. So I emailed her and it was like, can I be a high school student in your lab? And she was like, yeah, that's fine. Um, and I lived in the suburbs, so I only went like twice a month. So I really wasn't doing anything substantial, but it was just like a really cool experience. They let me work in a sterile hood. So that's my first experience with working in a biosafety cabinet. Um, and they'd get their samples from the hospital that they were attached to. So people who were at risk of an aortic aneurysm, they would like replace it with stent, but then they would get the um, tissue samples and then like process them down. So I got to do that too. And I was like, oh my God, this is so cool.
That is so
Um,
cool.
yeah. And I realized that, like, okay, the lady who runs the lab, she has a PhD. A lot of the people that get to like design projects, they have PhDs. Okay, I got to learn more about what a PhD is, what I want, like, how that would go. And at the time, I really liked biology and I really liked chemistry. And then I learned that biochemistry was a thing. And I was like, that's great. I love that. So I majored in biochemistry in undergrad because of that. I worked in a yeast lab in undergrad, so very different um sort of like transition, but I still got to do a lot of the stuff. I made a fusion protein to detect RNA DNA hybrids. So that was really cool. I eventually got a paper out of it. It got published like halfway through my PhD. But I thought that was still really cool. And now I'm here. No, I forgot. I did a post-bac program after I finished
Oh, nice.
my undergrad. So that was the NIH Prep post-bac program, which does not exist anymore. But it's for disadvantaged students that want to get a PhD. Um, so they put you in this program. You get to do full-time research in a lab. And I had generally expressed that I wanted to go back to working with like human samples. And the director of the program, her name is Niket Blake. I love her. She's the dean of the graduate students at UCSF now, so she doesn't run this program anymore. But she was like, I think that you should go into an immunology lab. I think you should do immunology. I was like, okay, but I'd never taken an immunology class. I had a biochemistry bachelor's, like I didn't do anything immunology. So it was a bit of a learning curve. I had to do a lot of reading and I ended up working in a B cell lab. And I thought that was so cool. I loved it so much. I was like, I get to work with like, I wasn't working with human cells because we were working in mice. And I learned that I did not like working in mice, which is fine. Um, but it was still like way more translational research that I wanted to get back to. And I thought that
Right.
was so cool. So I stayed with immunology. Even though I'm getting a biochemistry PhD, I still do immunology research. It's just
Gotcha.
in a biochemistry aspect, if that makes sense.
Yeah. Yeah.
So, yeah. So that's how I got here. And that's how I'm doing my PhD now.
That is amazing. It's so funny. Well, yeah, I was just talking to our, you know, my previous podcast guest, just about, she studies type 1 diabetes. So she's working on creating kind of, like you said, autologous B cells, like beta cells, I'm sorry, for transplanting back into people with type 1 diabetes, so putting cells back into them that can produce insulin. But, right, they have to make sure that the cells aren't attacked by the immune system. And so we're talking a little bit about immunology and just like how complex. Like she was just saying,
Yes.
like, of all the fields of biology, immunology is probably like one of the more complicated ones. Do you have like do you agree with that? And then
Yeah.
like, why is that? I guess, like, I was thinking, like, it's just like your immune system is basically evolved to be very diverse, to be able to learn, to, like, be very complicated in order yeah, be basically like an intelligent system, I guess, in a way, like, so that there's just so many parts to it. But, yeah, what's your perspective, I guess, studying immunology?
I would definitely say that it's super complicated. There's a reason why most cells only like study one cell type. So like we are an NK cell lab. I worked in a B cell lab before, and it's like sometimes you look at the intersection between like other cells that interact. So like the B cell lab had some T cell interactions. So we would look at proteins that were expressed on T cells, but it was mainly focused on the B cell effect because there's just so many things that you can look into. So
Right.
there's these things called CDs, which is cluster of differentiation. So, like I said, CD16 is one of those, one of the proteins that I'm looking at NK cells. There's hundreds of CDs on these immune cells, and they all interact with different immune cells and then all have different responses. There's cytokines that all interact with different immune cells, and depending on the immune cell, it might have a different response. And then the immune cells interact with each other, and depending on what point of the cell development that you're at, then that response can also be different. And then you're also talking about the process of the cell development itself, and that can also be really complicated and can also, there's like so many touch points in the process of like developing immune cells and immune cells interacting with each other. And then
Right.
there's cytokines. So immune cells will secrete cytokines based on what environment they're experiencing, what pathogens they've interacted with, and then that sends alarm signals to the other immune cells, and then they act accordingly. There's different kinds of cytokines that can be an activating or inhibitory based on whichever immune cell it's interacting with, or the greater like microenvironment, especially when it comes to tumors, like that gets really complicated. So there's just a lot to work with that you can't just study all of it. You get what I
Right.
mean?
Right. Yeah, it's like, I mean, it sounds like a brain almost. I mean, like a little mini.
Yeah.
Yeah. Right. Because it's like so many things communicating and happening together, which is, yeah, why your immune system is so not diverse, or was it so, like, adaptable, in
Yes.
a way of, like, it has to protect you from things that humans have seen in the past, but that also that we haven't seen, and
Mhm.
things in the future, and cancer that's gonna come from your own body, but still has some markers of being cancer. It's just your body has to figure out what those are, right?
Mhm.
Yeah. That's super cool.
Thank you.
Yeah. What's your, yeah, I don't know, what's, like, a challenge in your field, I guess, or in your study right now in your PhD, and how are you kind of overcoming that?
So let me think. I guess the challenge is like I'm doing my full project myself, and it my experiments take a really long time. So isolating my natural killer cells, probably isolating and then staining for the purity of the isolation, probably takes me about 6 hours. Um, to do my ADCC assay, where I add like I pre-treated my NK cells and then I do my treatment, that probably takes me, when everything is said and done, another 7 hours. And then I run flow cytometry to analyze those results. And that doesn't take me as long, but it's still like the machine is in a different building, so I have to go over there and I got to run everything. I've had to repeat this assay a lot because NK cells, the protein that I'm looking at, they also have these things called glycans on them, so they're sugars. And these sugar chains vary from cell to cell, and it can also impact binding. They vary from cell to cell inside of people, and they also vary from person to person. So I've had to do a lot of replicates. So I've probably done this. I've had like at this point almost like 40 human samples that I've like singularly processed and done all of the assays for. And that's a lot, especially because like I've done all of this treatment. So it's like I get my cells on Tuesday. So I get them tomorrow when I do this. And then I do my isolation. Then I run flow the next day on Wednesday. And then on Thursday, I do my ADCC assay. And then on Friday, I run flow again. And because they're primary human cells, I have to do them so quickly in sequential because at some point the cells will start dying. And
Uh,
if I mess something up in that process, I've lost the whole week. So that gets really frustrating, but I think I've done it so many times that I've got it down to like a rhythm. I just need to be like really focused. So like it would have been really cool to show you that too, but I can't because I will ruin it if I do that.
right. No, absolutely. Yeah, why do you, I guess, do it on so many human replicas? Are you looking to kind of standardize it in some way? Or are you looking at, like, how you would need to treat cells differently based on these different glycans it would be, or, on the different patient cell in the different patient cells.
I want it to be consistent results that I'm seeing. So like there's also two different halotypes of CD16A at amino acid 158. It can either be a valine or a phenylalanine, and that can also affect the binding affinity for ADCC. Um, in addition to just NK cells being like wildly different. Sometimes they'll get samples and they won't do ADCC at all. Sometimes they'll do a ton of ADCC. Sometimes they'll do a little bit of ADCC. Like it just varies so much based on the specific donor that the larger my sample size is, the more the more faith you can have in the results that I'm having.
Gotcha. That's really interesting. I mean, so like ultimately, like if we were able to create, well, like you said, like these, personalized essentially treatments for people's NK cells to put it back in so they could attack their cancer cells, like would those therapies have to be, would the like drug treatment of the NK cells have to be personalized for the patient? Or are you kind of just looking at like trying to find a protocol that would work for many different across many different patients based on, yeah, you've looked at many, many different cells. You're like, okay, this one kind of works across all of them, or
I'm
enough
hoping
of.
that it'll work across many different cells, but usually I've been seeing that if they don't do ADCC, then like there's nothing that I can do. Or they'll do like lower levels of ADCC, or they do like really high levels of ADCC, and that just depends on like the person. Um, and I can still, if they do ADCC, there's something that I can do, but sometimes like I'll get results that go one way, and then I'll do it again, and I get results that go another way. So like gathering as much information as possible from all of these different people, because there's like, we know that the allotype variation can impact it. We know that the glycans can impact it, but there are other ways that we don't know of that could possibly impact it. So as long as I have the samples and I've looked at like the stuff that I do know, um, it could just be like there's some protein, there's some function that like people haven't looked at yet that I don't know about. Or maybe someone has looked at it yet and then I just don't know about it, that might impact the variability. So if I'm seeing that across all of these NK cell donors, even though they are variable, I'm seeing this consistent effect. I know, okay, that's a real thing that I'm seeing.
Right, right. Yeah, it's so interesting and crazy. Like, why would some NK cells not do ADCC? Like, is it just you happen to get the wrong natural killer cells, or like,
I honestly don't know. I Someone might know. I might not have found the paper. I can't say that we
right,
don't know,
right,
because there
right,
are papers that I haven't read out
right,
there for
right.
sure, but I don't know.
Yeah. Yeah, I mean, it's just so interesting when we talk about, I think that's right in general, in terms of science communication, health communication messaging about, scientists are working on, right, cures for cancer. Um, it's just so, maybe maybe people are starting to realize, but I think it's not, people aren't aware enough of just like how much variability there is in this, and so it's like creating treatments for cancer. I mean, yeah, I think we're moving towards that like personalized, like looking at the individual patient, because there's so much that could be different, in one patient that has this specific cancer to another that has that same cancer, but they're going to respond very differently maybe to the same drug.
Yes.
Yeah. Awesome. Well, are your cells like already spun down and they're just like
Yeah, no, they've been
resting there?
sitting in there. I was like, we've got a flow going on. I don't
Yeah,
want
I got yeah. Oh, yeah, I don't wanna hurt your cells. Can they sit there for a
No, they're
while?
okay.
Gotcha.
I'll
I mean, I've always wondered, and it's silly, but it's like in, like, pipetting or, transferring the cells, to spinning them down or that kinda thing. I mean, do you damage cells at all and just in the process of doing that? But it's like most of them are fine? Like,
It's a thing that can definitely happen. So some cells are more sensitive than others. These ones are pretty hardy. So I, at least I don't think that I've been damaging any of them.
right, right.
but it's definitely something that can happen.
Right. I worked in labs where people are working with DNA and they actually, for like processing the DNA and sequencing it, they actually do want to shear it. So like cut it up
Mhm.
for sequencing, but like to different tuning it to different sizes. And that'll be like they'll kind of just like mechanically, I mean, I guess one way is just like pipetting really, really quickly back and
Mhm.
forth to kind of shear the DNA. So it's certainly, I guess it's like maybe having a gentle touch versus the like, you wouldn't want to like pipette up and down a million times and like start shearing all the things.
Yeah, I hope I don't do that. So I'm gonna resuspend my pellet at the bottom. I'm gonna be gentle.
Yeah, so for, people who aren't aware, I know so much of the terminology, so I'm like, "Oh, yeah, of course."
Oh yeah.
So I forget too that, when you're spinning the cells down, I mean, you're essentially using, right, centrifugal forces, so
Yes.
that the heavier things in the media, the cells kinda come to the bottom, and then they're kinda all squeezed together, and that you need to, like, that pellet of all the cells right at the bottom of the tube, you're having to kinda get them to go back into suspension a little bit. Um, yeah.
Yes. Sorry. Yeah, so my cells will spin all the way at the bottom. The dead cells will stay with the old media.
Gotcha.
Um, so what day is this? It's the 27th. Okay. So I will resuspend my cells in the fresh media. So it's like, let's say you like your parents have left you at home for like 3 days and you're like a teenager and they leave you with like um a pantry full of food. You go through it in 3 days and you're like, "Oh my god, I'm so hungry." Then your parents come by and they refill the pantry with the food and then they leave again. That's basically what I'm doing. So I'm refreshing the food source of the cells by spitting them down, and then I take all of the dead cells out and then I add new food in. So now they have fresh media. You can tell that there's cells in there because it's a little bit cloudy. And
Yeah,
then I write down the date that I passage my cells and then the passage number. And
gotcha.
then that's basically it. So I'll put this back in the stationary incubator and then I'll clean up in here. I'll bleach these cells because I can't just throw them down the sink. Because they are cancer cells, so I have to bleach them and make sure that they're dead, and then I can pour them down the sink.
Gotcha.
And then I'll put everything else away.
For the B cells you're working with, do the cells, like, do they grow on the bottom of the dish, or do they just kinda stay floating around?
They stay floating around, so they grow in what's called suspension. And then the ones that grow on the bottom of the dish are called adherent. So they just stay in the media. If they grew on the plate, then I'd have to do a different protocol. You use something called trypsin, and then that's how you detach them from the plate, and then you
Right.
can spin them down. But these ones grow in suspension. I think all of our cells grow in suspension. So
Gotcha. Yeah, it's kinda crazy to yeah, to use cancer cells that you're just growing in a
yeah,
dish for your experiments. I mean, it's, like, normal to scientists, but it's also, like, that's kinda crazy.
it's one of those things where it's like, I'm so used to it that I don't think about it, but I know if I got like a 12-year-old in here and I started explaining what I did, they'd be like, "Oh my god, that's insane." They
Yeah.
don't have to the 12, just someone who doesn't do science
Right.
regularly.
Right. Can you talk about, like, in terms of the process of research that we start with maybe cell culture research, and then, like, to validate that further, if we're creating a drug or whatever, that's when we might move to animal models, or we might move to organoids or to, like, more complex systems, so we can validate, yeah, what happened in just a cell culture in a dish, which could be a little bit, right, different than how cancer might be growing in your body, I guess.
Yeah. So we usually start like, I guess the whole drug discovery process will start at the very beginning. Like you have like this panel of drugs. You're trying to see if it gets any hits against like this target. Usually it's some sort of protein or something like that. And you want to see like does it interact with this protein in a way that we want it to? And then from there, you could have like 10,000 different drugs. And from there, it narrows in. And by the end of the process, like after clinical trials, you have one drug that you know that works. So it's like a very time intensive process, probably takes like 10 to 20 years. So my part that I'm at right now is like after that you end up working in vitro, so in a dish, not inside of the body, not inside of organs or anything, but it's like outside of the body. So I'm doing all of my experiments on cells, and like you said, yeah, certain cell lines, after a while they might not be exactly where you want them to be. They might have some sort of variation just because you've been passaging them for a while and they are cancer cells. And that's why it's a step in the process and not like the end all be all of like, now that we know that this works, we can go do other stuff.
Right.
So usually from there, you have, out of the targets that you have that know, you know that they interact with this protein, then you put them in a dish, and then you're like, okay, um, how many of these drugs will end up doing the thing that we want it to do. So in this case, it would be kill cancer. How many of these drugs end up killing cancer specifically focused on this protein that we want it to do? And how efficient are they? And then you get a smaller subset of things. And then you'd either move on to preclinical models. So that could be organoids, that could be animals. And I know that like doing animal testing is a contentious subject, but at the
Right.
same time, you don't have anything that can replicate an entire organ system in the way that animals do
Right.
before we can get to humans. So it's really unfortunate that it's like that. Um, but we can move to organoids. So they're like, um, they're not necessarily organs. These are also things that we can grow in a dish, but they're like the line between like just cells and working in organs. They're like something, they're a little bit of both. They're not a complete organ,
Right.
but they're not just cells. They have like an environment and they work together in synergy. Um, so then if they work there, then you have a smaller subset that works there. You have a smaller subset that works in animals. And working with animals is really important because that's when you see like, okay, maybe it's killing cancer here. Maybe it's killing these cells, but then it's causing a tumor somewhere else. It's causing these other problems. All of our animals are dying. Like then you know, okay, we can't use this drug. We have to move on to a different drug. So then you have a smaller subset. And then that's when you start going into clinical trials. And then there's phases of clinical trials. There's phases 1 through 4. I'm taking a class on this right now, so it's like very fresh in my brain.
Nice. Nice.
Um, so you have 1 through 4. First phase is uh usually small group, healthy people, and they're just saying, okay, does this drug cause any bad side effects? If I give this to people, will it make them really sick? Hopefully not. And then if it does, they immediately pull out and they're like, we can't use this anymore.
Right.
Um, so that's phase 1. And then they're kind of seeing how much can we get away with giving, like how much of a dosage can we get away with giving to people? That's when they start figuring out the dosage. Phase 2 is usually with a larger group and it is sick people. And then that's when you start seeing, okay, what kind of dose range can we look at here in terms of effectiveness? And then phase 3, that's when we're getting to like thousands of people or tens of thousands of people. And this is where we have sick people with the disease and you have the treatment versus a placebo groups of people that might also be sick, but they're not using this new treatment. They're either using a different treatment or they're using something that doesn't work. Usually, from an ethical standpoint, I think they do use a different treatment that
Right.
already exists in the market and just not the new one. And then the patients, usually it's either single or double blind. So either the patients don't know what drug they're having, or the patients don't know what drug they're having, and the people giving them the drug also don't know what drug they're having, to rule out any sort of bias. So this is at this point when the drug works, this is when it starts getting like FDA clearance and stuff like that. And then stage 4 is called pharmacovigilance. And this is required testing after the drug goes to market. And it's usually like years to decades of, okay, this drug is safe. It's not causing any side effects in like the general, any like alarming side effects in the general population that we're seeing. So there's that. Sometimes people have an optional stage 5 where it's like they're monitoring for safety the entire time the drug is on the market, even past that required deadline of monitoring.
Yeah.
So that is the whole process.
Yeah, it's so interesting. I mean, it's just like I was having visions, right, of like the COVID vaccines and, and yeah, how people maybe aren't, if they're not aware of all those different phases, right? Like
Mhm.
just because a drug goes to market, I mean, usually that means it's right better than
Mhm.
nothing, but there, I mean, there
Yeah.
could still be issues that, and they need to also figure out like for who there are issues because
Mhm.
maybe for most people it's safe and for some they might have side effects or whatever. It's really interesting. I did see, I guess I was following another neuroscientist who did an Instagram post recently talking about how like they just FDA just kind of I mean, I need to go and look at the specific details, but that the FDA just made some kind of decision that would like make it so animal models weren't needed if you did kind of organoid or some of
Yes.
these other things, that it could go straight. And he was saying like this is, a good thing, a good development. But yeah, talking to a lot of scientists though, it seems like most scientists agree that like you said, that I mean the organoids are great, but and they're developing further, but maybe like are we really at that point where we can fully model
Mhm.
what it would look like in an organ to, like, not need animal models? Like, it seems like for a lot of things, we're maybe not there yet, but
Yeah. It's really unfortunate because like it would be nice, but it's like maybe you're trying to treat like a blood cancer and maybe you treated the blood cancer but now you have tumors in the brain and you wouldn't know that if you're using organoids you wouldn't know that if you're
right,
doing it in like dishes but you would know that in an animal model and you would know that in humans but you're trying to prevent that from happening in humans so I feel
right,
like it
right.
like opens up a different world of problems that because I'm not entirely sure if scientists were involved in the decision, but I'm going to assume probably not.
Uh,
Um, so I feel like it's just something that they might not be thinking about in a whole manner. But there are smarter scientists than me. Like I'm still doing my PhD.
no,
There
no,
are people who
yeah.
have PhDs and they've been scientists for like decades. But from my humble grad student opinion, I feel like that's something that we should not do.
No, I think yeah, I think you're absolutely right, and, like, I mean, I need to do more research on this, but, um, there was another story recently, I think it was covered in Science News about,
Mhm.
um, someone a young woman in China who was treated with a gene editing therapy, basically, and it was like I think it was kind of done on this kind of personal, like, one-on-one case, like a therapy created for her, but, like, there was some animal studies done that what the report didn't come out until they kind of already decided to move ahead with her, and it was essentially like yeah, essentially, like, there was kidney, liver, like, other effects of this therapy that ultimately did, end up killing her, but it was like from the animal models, it was obvious that these were there. But like you said, you unless you used a whole system like an animal to study that, you wouldn't see like different or effects in different organs or effects like across the whole holistic organism if you're just using an organoid. Yeah. So,
Mhm.
yeah. I guess you're hoping that your drug doesn't have those off-target effects in a different organ, but you don't really know until you
Yeah.
test it, yeah.
And it's not like, as far as I know, because we have a whole host of rules when it comes to working with animals. Like I personally don't work with animals anymore, but I know when I did work with mice, there's a whole host of rules of like, this is specifically how you're supposed to handle them. You can't handle them in any other way. If they're in pain, you can't just like be like, okay, I wanna unless you're like a pain researcher, you can't just be like,
Right,
well, I want to know how bad the pain is, like how far can this go? Like as soon as they're having any sort of like bad side effects, you're like, okay, gotta like they call it sacking, sacrificing this mice. You have to put down the mouse. You'd be like,
right,
okay. And if you want to get any data from that, maybe you can take like tissue samples, but you're not like torturing this mouse for fun, you know?
right, 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 right. Yeah, well, thank you so much for, yeah, showing us around the lab and talking about
Of
your
course.
research. Yeah, it's been super fun. I learned a lot. Obviously, like I said, immunology, just diving into it a little bit and realizing like it's such a cool field, but also like so
Mhm.
complicated, right? That we need a lot of different researchers working on a lot of different aspects of this to figure it out. But I don't know, just like wrapping up, is there anything you're really excited about for the future, I guess?
Um, Probably finishing my PhD.
Yeah, yeah,
I've
you're
got
close, right?
Yeah,
Yeah.
yeah. I'm about to start my 5th year, so I've got like little less than a year to go. Um, I think it's been like a very um I've learned a lot during the process of my PhD for sure, but like after a while you're just like, okay, can we get this thing rolling?
Yeah. Yeah, do you think are you wanting to stay in research? Are you wanting to stay,
Yeah,
yeah, like in academia or kinda industry research or
I think I'll go to industry research for like a number of reasons that I won't necessarily get into,
Yeah,
but I think it's just one of the reasons is that I think it's really cool that like I can see the fruits of my labor, the way in academia you can study something that's like really interesting and really important, but it's not something that will necessarily get to market or get to
right,
something where it's like this is a treatment that'll work for quite some time, maybe even your whole career, because they're like, okay, this isn't important enough to look at.
right.
In a way that industry, although if I still work in preclinical, it might be a lot of the same thing, but like I can be the person who like brought a drug like Keytruda to market through the preclinical work, and I think that's really cool.
Yeah, that's amazing. Yeah, I agree with you. Yeah, just being able to see that, knowing that you're working on, I mean, I admire people so much who do basic science research
Mhm.
and really just asking questions and getting curious, and you might come up with something that later down the line really helps people, maybe not, but you're kind of like weeding through those things. I always do like to kind of need I'm like a person who needs that validation and that kind of instant gratification of knowing like that this mattered.
Mm.
But so that's really cool. I think that, yeah, it must be really exciting working in pharma or in like industry right now, just with the amount of like technologies that we have. I mean, from like
Mhm.
organoids to like AI to being able to like look at the complexities of the immune system maybe and modeling them down to understand them better. It seems like a really exciting time for that.
Yeah, I think so too. We've really gotten into like this might be a little bit old news, but I know like spatial transcriptomics and spatial
Yeah.
proteomics is really cool because it's like, okay, we can see like the proteins or the mRNA that these cells are making at this specific tissue in
Yeah.
this specific area and see how that factors into everything else. And it makes a ton of like kind of confusing graphs because they have the UMAP axes, which I have beef with the UMAP axes, but like I understand why it's
Yes.
set up the way it is. And I
Yeah.
think that can be really cool as well. Like that sort of deep dive into results on a cell by cell basis.
Yeah. That's amazing. Awesome, Zainab. Well, thank you
Mm.
so much. It's been a pleasure.
Of course. Thank you for
Yeah.
having me.